Last time we spoke of the contractile cardiomyocytes and how they communicated their action potential from cell to cell. What I want to do here is take a step back and look at the organization of cardiac tissue, and I'm going to compare it to the organization of skeletal tissue because there are important similarities, but there are also important differences as well. So I have a cartoon over here, and that's going to be skeletal muscle. And then over here is going to be cardiac muscle. So what happens? During an action potential in the cardiac muscle, you have a depolarization, goes into the cardiac muscle or cardiac tissue via T-tubules, leading to an opening of dihydropyridine receptors, which are basically voltage-sensitive calcium channels, and these are the L-type variety here. So let's take those words and let's connect them to the cartoon below. So what we have going on here, cardiac, there's a T-tubule here. So what we're having is we're having a depolarization wave that is going to be communicating through the T-tubules, and they are then in turn going to be opening up dihydropyridine receptors or L-type calcium channels. So we had a T-tubule and we saw a similar motif having to do with skeletal muscle. So here is the T-tubule on the skeletal muscle. T-tubule, how about that T-tubule there? So here he is right there compared to the T-tubule there. So one of the differences you see right off the bat is that the T-tubule on the cardiac muscle is much, much larger than the T-tubules in the skeletal muscle. Now you will recall back in terms of how the skeletal muscle worked was that you had a wave, a depolarization wave going into the T-tubule. Again, it originates on the surface and then goes in. And then you have these, little dihydropyridine receptors are sitting on the T-tubule surface, and they're going to, again, as the T-tubule surface depolarizes, it's going to open up those dihydropyridine receptors. And you recall here is go different color here maybe, let's go blue, and we'll do maybe here. So remember you have your lateral sacs here, terminal cisterna, and then of course you got your network of your sarcoplasmic reticulum there. And you'll recall the way it worked in the skeletal muscle. You had a T-tubule here, and then let's say we have let's see, I went the wrong color there, so let me back up on there and try this again. Got that, and we'll go red just to be consistent. So I'm going to have a dihydropyridine receptor there, or an L calcium channel. Okay, and so the wave of depolarization is going to open that channel up, but also what we have in the skeletal muscle, remember we have a lateral sac here. And we'll just go there for my lateral sac or my terminus cisterna. So we'll go TC. And then sitting on that is going to be another type of receptor, and that's going to be my RYR1 receptor, RYR1, and that it is physically connected to the L-type calcium channel on the surface of the T-tubule. And another term for the RYR or ryanodine receptor is going to be foot protein. You'll see that sometimes referred to that way. And so this channel this L-type calcium channel or the dihydropyridine receptor opens up, and that because it is physically connected to the RYR1 receptor, the change in shape of the L-type calcium channel will also change the shape of the RYR1, and it will open it up, allowing then calcium to flow out of the lateral sacs, and of course you have your filaments over here, allowing the calcium to do what they do regarding the myosin and the actin interaction. So that's how that works. And you might have just a little bit of maybe a calcium flowing in here that might influence the activity of that RYR1 receptor, opening up, allowing calcium in. So that's kind of how we saw that, again, relative to the T-tubule here. So we're going to find out things are a little different when it comes to the cardiac muscle, and I got a prettier diagram that shows you that on the next page. All right, so over here in the cardiac, you have a T-tubule, and the T-tubule also is going to have these L-type calcium channels or the dihydropyridine receptors on them. All right, so you're going to have that, and we'll go red here. All right, and then I'm going to have this sarcoplasmic reticulum, and so the sarcoplasmic reticulum here is going to be this network there. So right off the bat, what you see, there's a difference. You don't have these robust lateral sacs in the cardiac muscle like you do the skeletal muscle. The skeletal muscle has a lot more to the sarcoplasmic reticulum compared to the cardiac muscle. So the cardiac muscle, the sarcoplasmic reticulum is rather exiguous relative to the sarcoplasmic reticulum that you have in the skeletal muscle. Very scanty, very skinny, but again, it's enough to do the job. So that's one thing you're going to want to note. So what you have here, again, just cartooning it out real quickly, is I'm going to have the sarcoplasmic reticulum. I'm just going to say this is this right here. And then I'm going to have, I went green on this one, so we'll go green again. So there, I'm going to have an RYR2. RYR2, a ranodine receptor. So this could be sitting there. Now, one of the differences is for the cardiac muscle, the dihydropyridine receptor is not physically connected to the ranodine receptor. It's very close, but it's not connected. And so again, in this situation here, you'll have a depolarizing wave. It's going to allow calcium to flow in, and the calcium may interact a little bit or will interact with that RYR1 receptor, and as that calcium flows in there, it's going to open, then allowing calcium then to flow out, and of course you have your actin and myosin over here to do what they do. So that's kind of a simple cartoon of how they're a little different. Again, a much more detailed cartoon on the next page for this. So you have that. So again, you both have T-tubules. They both have sarcoplasmic reticulum, except the sarcoplasmic reticulum is much more well developed in the skeletal muscle fiber compared to a cardiac muscle cell. Another thing you'll note, they're both striated, so you have your I bands, A bands, etc. The arrangement or placement, though, is different. So you'll recall on the T-tubule that you have, it's going to be in line with the AI boundary. Okay, so there's the AI right there, the AI interface there, and it is perfectly lined up with the T-tubule. Here's another AI band, and again, T-tubule. Well, if you look over here in the cardiac, the AI is actually right there. So it is not lined up with the T-tubule. So the T-tubule is actually in the cardiac muscle lined up with the Z-disc. Whereas the Z-disc over here is not lined up with the T-tubule. So yet another distinction in terms of how they're just a little bit different. Okay, and these are common questions you'll get on exams and such. So again, you want to be familiar with basically how things are laid out. Okay, so going back here into the verbiage. Again, contrasting, voltage-gated dihydropyridine receptors, T-tubules do not mechanically, got all that already said, and it's kind of going down through here. I've already said all that as well. Let's see, the T-tubule is about 5 times as large as the T-tubules in the skeletal muscle, so it gives you a number there to pay attention to. Again, the Z discs, I've already talked about that. AI junction, I already talked about that. And we'll see on the next page, calcium is removed from the cytosol, put back in the extracellular fluid by a NCX exchanger. Again, more on that later. And a calcium ATPase pump. Again, we'll see that on the next page as well. The other thing that I want to point out that's not in the diagram, but is still very useful to know, is with the T-tubules in the skeletal muscle, they're all kind of just in this orientation here. The T-tubules in the cardiac muscles, it looks like they're in this orientation here, but they will actually come in and then turn and go longitudinally. So they'll come in and they'll run this way. I don't know how well you can see that. Maybe I'll go with the red. The T-tubule will come in, and then they'll run longitudinally here, and that makes it a little more efficient in terms of the action potential wave going down the T-tubule and exposing itself to more or a greater part of the skeletal muscle tissue, the cardiac muscle tissue itself. So another difference, again, is the T-tubules both run latitudinally and longitudinally in the cardiac muscle, whereas the skeletal muscle is just kind of more lateral. So again, another difference there. Okay, so let's go into and look at the cartoon diagram of a cardiac muscle and just see kind of how things are working. Again, oversimplified diagram, but it's enough for us to have the general idea of how things are working. So we're going to say here that this is going to be the T-tubule, okay? So we'll label it as such, T-tubule, okay? So the action potential then is going to be, going down in that direction there. And so it is going to be then opening up a L-type calcium channel. That's my dihydropyridine receptor. Right there, again, L calcium channel. It's going to open up, allowing calcium to flow in. And the main job of the calcium flowing in is to make contact or interact with the RYR2 receptor, my ryanodine receptor type 2. Again, skeletal's type 1, cardiac is type 2. So this calcium here is actually acting as a trigger for that RYR2, and then it will open, and the vast majority of the calcium that is going to interact with the filaments down here is actually going to come from the sarcoplasmic reticulum. Some could still come from the outside, okay, so it does get into the act as well, but most of it comes from the sarcoplasmic reticulum. So chain of events, you have your action potential going down T-tubule, opening up the hydropyridine receptor, lets a little calcium in. Calcium then is going to trigger the RYR2. It will open, calcium flows out. And now we have sources for calcium to be in the vicinity of the filament so that the actin and the myosin then can interact. So you have that. So let's see what we have here. Make sure I haven't left anything out. You got that. Okay, got that. And L channels, got that. Another term for the calcium as it interacts with the RYR2, sometimes you'll see that called sparking. So that little bit of calcium that comes in will spark the RYR2 to open up, allowing more calcium to flow out. What's interesting in cardiac tissue, and the same thing really is true for skeletal as well in terms of the RYR1, is that you can have spontaneous sparking. And you don't want that. It would just open up. And when you have that in cardiac muscle, that could be a problem as far as arrhythmias is concerned, because that's not what you want. Another thing that can go on is that RYR2, if it spontaneously sparks, it can also lead to nearby RYR2s to spark as well. So you start a chain reaction, you get the chain reaction going, and all of a sudden you have an arrhythmia that you don't want. These RYR2 receptors can be regulated by second messenger systems and mechanisms such, calmodulin kinase, which can phosphorylate the RYR2 receptor. Again, I'll make this point over and over again, that all of these major units, functional units, whether it's a receptor or a pump, is subject to regulation by various cellular processes, whether it be by G proteins or phosphorylation or dephosphorylation, etc. So all of these can be controlled and modulated. When the RYR2 receptor is phosphorylated to an increasing degree, that actually can make that channel be more sensitive and allow more calcium in. So an example, RYR2 receptors are responsive to caffeine, norepinephrine, epinephrine, again, probably involving some phosphorylation process where it makes it easier to open up and then obviously affects the functioning of the heart itself. So you have this flow of calcium coming in and doing its thing. Then the question becomes, how do you reset the whole system? Because you can't just always have calcium flowing out. You have to have a way of resetting the clock, so to speak. So multiple things that are going on here. One of them is we have a Circa pump, and this is Circa type 2. Okay, smooth endoplasmic reticulum, calcium. So you see where the Circa is coming from, type 2 in this case here. And it is an ATPase pump. And its job is to take that calcium that was flooding into the intermediate, to the microfilaments here, actin, myosin, and return it back to the sarcoplasmic reticulum. Again, that makes sense. I mean, you got to be able to pump that in there. It's going to take energy to do it for, again, the next cycle. So you have that. Now, you also need to be clearing out the calcium from the inside and pushing it back out into the extracellular fluid. And so one of the things that will do this for you is going to be a calcium ATPase pump. I just got through telling you that the Circa 2 is a calcium ATPase pump. They're not the same pump. Okay, you got many, many different forms of calcium ATPase pumps, and what I'm telling you is that this ATPase pump is not identical to the Circa 2, although they're both calcium ATPase pumps. So here's another way that you can get calcium out of the cytosol or the cardiomyocyte back into the extracellular fluid. Another pump that we've already talked about earlier was the NCX pump. That's an exchange pump. Again, it's going to pull in this case, pull in 3 sodiums to get one calcium back out. Again, the theme is how do you clear the calcium out from the cytosol? And of course, it is using the concentration gradient of the sodium to supply the energy to push that calcium against its concentration gradient and its electrical gradient. Of course, we have to manage the sodium and the potassium, and what's not in here is all the potassium channels. Again, that makes a diagram probably overly busy, but you can understand there are a lot of potassium channels coming in here as well, as we saw on the prior page, but that's just kind of left out of the diagram here, and that's okay. You understand that they're there. But we still have to have a way of managing the potassium, managing the calcium. Of course, we have our standard sodium, potassium ATPase pump that is helping that as well. Okay, so that's kind of a nice scheme diagram, oversimplified for sure, but just gives you a general sense of the movement of the ions and how you regulate those and how you get the calcium into the sliding filaments to get them to do what they do. And then how do you reset, return the ions where they need to be so that you can go through then the next cycle. So again, this is what's happening in terms of a cardiomyocyte. Now again, don't lose sight of you have that cardiomyocyte, but then you have to communicate that action potential to the next one, and that's where we're back into the various types of ways of shifting electrolytes from one cardiomyocyte to another cardiomyocyte, and we talked about that last time, so I'll not rehash that. Okay, so last point on this page that I want to mention is, again, this is kind of a clinical point. It's kind of cool. catecholinergic polymorphic ventricular tachycardia. That's a mouthful. Well, tachycardia, you know, abnormal rate of a beating of the heart. So it's racing, it's beating very quickly. Ventricular, telling you where it is happening, because you could have tachycardia of the atria or tachycardia of the ventricle. So in this case, it's going to be the ventricle itself. Polymorphic, in other words, the wave shapes are very different, and we'll talk more about that later. In a catecholinergic, again, catecholamines, what are we talking about here? Talking about epinephrine, norepinephrine, you know, theoretically dopamine, etc. So you're putting all these together, just looking at the name, you can get a sense that what I'm having here is I'm having a type of tachycardia of the ventricle, and it is being induced by a catecholamine, epinephrine, and norepinephrine. So what we have here, this is actually a genetic defect. It's an RYR2 genetic defect. This is autosomal dominant. About 1 in 10,000 will have it. And it comprises about 15% of sudden unexplained cardiac deaths are caused by this. Typically, no symptoms. You don't know you have the genetic defect until most of the time it kills you, which is very sad. But what happens is during exercise, as you know, your norepinephrine and epinephrine can increase for a variety of reasons. You're wanting to, again, speed things up because you're exercising, etc., so that is not surprising at all. But if you have this defect, it's overly sensitive to norepinephrine and epinephrine, and it can cause an abnormal amount of calcium sparking. Okay, going back to the RYR2, again, there's something wrong with it. So now it's oversensitive to presence of calcium, particularly if you're having a catecholamine, norepinephrine or epinephrine influencing the RYR2, and we're going to see later that when the heart sees a sympathetic stimulation, again, epinephrine, norepinephrine, there are cellular changes within the cardiomyocytes, and so you're going to have changes in the RYR2 receptor. You're going to have changes in many other things as well. But what I'm saying is that with this genetic defect, you can have a change that in response to exercise, which is going to come through epinephrine, norepinephrine. Something goes sideways. These things start sparking uncontrollably, and now you're going into possibly ventricular fibrillation. So the tachycardia means beating very fast. And you can survive tachycardia, but many times, as we'll find out later, tachycardia can lead to fibrillation, where you have a complete discoordination of the individual cardiomyocytes contracting. So, and if they're not coordinated, then it can't generate pressure in the ventricle. Therefore, you're not pumping blood. If you're not pumping blood, blood pressure goes down, and then, of course, you die. So, pretty interesting, pretty serious stuff. Sometimes the symptoms are there, okay? A lot of times they're not. The first symptom is death, but sometimes, if you get lucky, you'll have some symptoms as you're aware of this. When they are present, again, tachycardia, lightheadedness. Now, why would you have lightheadedness? You'd have lightheadedness because if you have a certain degree of tachycardia, that drops down the efficiency of the heart pump, the pumping action of the heart anyway, because if you're beating too fast, the heart is not able to relax long enough to get blood back into the ventricle for the next push. And so you can certainly reach a point where the ventricle is contracting so fast, so quickly, that it doesn't have time to refill. Then that then leads to a compromise in cardiac output being how much blood goes from the heart to the rest of the body. And then if you're compromising cardiac output, you're compromising blood pressure, you get lightheaded. Dizziness or syncope, same sort of thing. Well, they're not the same thing, but similar. Also, this particular pathology can cause after depolarizations. We'll talk about that later, but just quickly here, recall last time we have a action potential of a cardiomyocyte, something like that. Okay, normally it would go like that, right? Well, an after polarization is where, let's see if I can get up. Okay, well, I can still do this. It's going to go down, and it's going to go down like that. Okay, but once it gets beyond its refractory period, now all of a sudden now you have a series of action potentials before it reaches baseline. Again, we'll elaborate on that later, but that's not a good sign when you have this type of after depolarizations. This particular genetic defect, if you have symptoms, it's going to be typically going to be approaching or going to be appearing a child between 6, 7, 8, 9 years old, somewhere along in there. And again, that's when these problems can arise. Okay, so that's kind of a whirlwind tour of what's going on as far as movement of calcium and then managing all the ions for a cardiac muscle. All right, so we will pick back up next time.